study of life cycle of one of the mushroom. Name of that mushroom is Agaricus bisporus. So one of the edible mushroom widely cultivated throughout the world and commonly or popularly known as a button mushroom. So how its life cycle is, that we are going to see during this discussion. So Agaricus, one of the edible mushroom, is saprophytic fungus. Grows on soil humus, decaying litter, on forest floors, in the fields and lawns, or on wood logs and manure piles. It can grow best in moist and shady places, and during rainy season one can see this mushroom at many places. It is cosmopolitan in its distribution. About 17 species of Agaricus have been reported from the India. Out of that, Agaricus campestris is one of it. One of the poisonous species of the Agaricus is Agaricus xanthoderma. Now we will see structure of thallus. So first, viridium mycelium. So it originates from primary mycelium. After fusion of hyphae of two opposite strains, the nucleus of one hypha migrates to other and later gives rise to binucleate secondary mycelium. So this happens inside the soil. And this phase, which is under soil, it is long-lived and abundant one. The secondary mycelium, after formation of, just see again, two opposite strains nuclei from two opposite strain hypha. For example, just see how it happens. Suppose this is one monochromatic hypha having one nucleus. This is a positive strain, and another monochromatic hypha. It is of negative strain. Such two hyphae under the soil, they come together, and like this, two nuclei, one from positive and one from negative, they come together and dikaryon is formed. The structure, this cell, it is called as a dikaryon. And further, it divide and redivide mitotically, and this dikaryotic nature is maintained from here onward. So call it as a primary hypelium, primary mycelium here, and from here onward call it as a secondary mycelium. So secondary mycelium, it grows extensively under the soil and becomes organized into special tissue to form a fruit body named as a basidiocarp. So some of the stages of development are shown in this figure, how basidiocarp development takes place. So under soil you can see the union of the primary and secondary mycelium, primary mycelium to form secondary mycelium, then formation of the next stages like button. You can see it over here. So formation of fruit body, that is the basidiocarp, that starts. Fruit bodies appear like umbrella. The above ground portion of fruit body that looks like a umbrella, and it is made up of dikaryotic hyphae. See here a structure of basidiocarp, mature basidiocarp you will see here. So this is pilus, these are the gills, then this structure is a stipe or stalk, and this is the annulus. See here one more picture of mature basidiocarp. So rhizomorph-like body, it is responsible for the formation of stipe. Stipe elongates and it bears the upper portion, which is called as a pilus, and pilus on its underside it bears gills. So arrangement of gills you can observe over here, and the detached portion of velum, it is here, and call it as a when pilus expands, there is detachment of the velum from the stipe, and the remaining portion it is called as a annulus. So mature basidiocarp, it is a fruiting body of Agaricus bisporus formed as a result of sexual reproduction is in front of you. So dikaryotic hyphae, when these hyphae, they are under the soil, when they come near to the soil surface and when conditions are favorable, these hyphae, they intertwine and forms a structure named as a rhizomorph, and this rhizomorph further develops into the mature basidiocarp. Now we will see how reproduction in Agaricus takes place. Three types they are there: vegetative, asexual and sexual as usual. So vegetative reproduction mycelium through mycelium. So asexual reproduction by formation of chlamydospores. Chlamydospores germinate and give rise to the hyphae, and further the mature basidiocarp development take place. Another asexually reproducing body, that is idea. So it may be also formed under conditions, certain conditions which are responsible for formation of idea, and this idea can also form the fruit body. Further developments they occur over there. Now related with the sexual reproduction. See here, you can see the structure of gills. You can see over here, this is the basidiocarp, also called as a sporocarp. This is the region of pilus. This is hymeneum, long gill or full length gill, and this is short gill. So further details here related to the structure of gill, where you can see the central trauma region, then on both sides subhymeneum, which bear the portion named as a hymeneum, and in between there are paraphyses, the sterile structures named as paraphyses. So hymeneum, it contains the structures like this, and these are, or this is basidium. So basidium, this one is basidium, it bears basidiospores. So one basidium, it bears 4 basidiospores. Out of that, 2 are of positive strain and 2 are of negative strain. So this is in brief related with the structure of gill. Overall, again one can see here how the gills are and what are the different parts anatomically of the gill portion. So sexual reproduction, it is somatogamous or pseudogamous. Sex organs are completely absent and their function has been taken over by the somatic hyphae, which are heterothallic. But in case of Agaricus, bisporus here, somatic hyphae are homothallic, means present in one and the same thallus. So in sexual reproduction, here one can say that plasmogamy followed by karyogamy, which is again followed by the meiosis. So sexual reproduction takes place after going through plasmogamy occurs first, then karyogamy, and next that is meiosis. So plasmogamy is the first step of sexual reproduction of Agaricus bisporus. Here, vegetative hyphae, which are uninucleate haploid cells, forms mycelia of opposite strains and come in contact and fuse. Each of such fusion results into binucleate cell, and we call it as a dikaryon. That I have told you by sketching the figure how dikaryon is formed. This dikaryotic cell then divide and redivide successively and mitotically to form binucleate mycelium, also called as a dikaryotic mycelium. So conversion of monokaryotic mycelium of negative strain into dikaryotic mycelium having two nuclei in each cell, and further this dikaryotic mycelium is perennial and it produces characteristic fruit body responsible for production of characteristic fruit body, that is the basidiocarp of this mushroom. Year after year this thing happens, production of basidiocarp from this subterranean dikaryotic mycelium. Now karyogamy. After plasmogamy, what happens in plasmogamy? Fusion of two different strain nuclei which are monokaryotic. Now karyogamy. In dikaryon, remember, two nuclei never fuse. So fusion of cytoplasm only, nuclei do not fuse, they remain side by side, and after that karyogamy. So second step in sexual reproduction, that is karyogamy. It takes place when formation of young basidium occur in the gills. So where fusion of two nuclei of dikaryon takes place in the young basidium, and the process is named as a karyogamy, or phenomenon is named as a karyogamy. So now young basidium is having fusion of two nuclei, then meiosis. This is third and last step of sexual reproduction, wherein basidium, prior to formation of basidiospores, this stage occur. So karyogamy is immediately followed by meiosis. So this diploid nucleus undergoes meiosis and four haploid basidiospores are formed. Out of that, two are of positive strain and two are of negative strain. We have seen how basidiocarp look. Now once again we will see the structure of basidiocarp, mature basidiocarp. So the subterranean mycelium, that is dikaryotic mycelium, it intertwines and forms a structure named as a rhizomarp. So rhizomarp absorbs sufficient food material and produces fruiting body, which are small in size and remain underground at first. These tiny pinhead structures come above the soil under favorable conditions, for example, after rain or after getting enough moisture which from the soil. So these pinheads, they come above, and these are called primordia of basidiocarp. These primordia, they enlarge, they becomes round or ovoid and represent the button stage of basidiocarp. So we have seen somewhere here, these are the buttons. See, button stage of basidiocarp. If longitudinal section of button stage is taken, then one can see the structures somewhat like this. These are some of the stages of development of basidiocarp. This is the first, second, then third, and the fourth. And we can have the mature basidiocarp like this one. So it is also called as a sporocarp. So this fruit body is divided into three parts like stipe, then pilus and annulus. Stipe is basal part of basidiocarp. Pilus is its top mature pilus is of 5 to 12.5 centimeter in diameter. Structure of gill also we have seen. So long gills, half length gills, quarter length gills. So these things, they represent the structure of gill. And if cross section is taken, that is of pilus, how the arrangement of gill is, that you can see. This is half length gill, this is full length gill, this is the stipe, like that. So in transverse section, the gill shows trama. So this trama, which is the middle part of the gill, is made up of loosely arranged intron mass of plectenchyma or plectenchymatous tissue of long slender hyphae. These hyphae, they run more or less longitudinally in this region. Then subhymenium region. After trama, you take the region that is subhymenium, or this region, this one. So in this region, the hyphae of trauma, they curve outward towards each surface of the gill, and they end in small diametric cells forming compact layer of subhymenium. So this is a compact layer of subhymenium. Now hymenium. So this layer, hymenium, outermost layer lies on surface of subhymenium covering both sides of gill. Some branches emerge out at right angle to the subhymenium and develop a palisade-like layer consisting of basidia, which are fertile. And they bear sterile hair-like structures called as paraphyses. So in between fertile basidia, sterile paraphyses are also observed. And these basidia, they bear basidiospores. So see here, you can see basidiospores. Here, basidiospores. Now, these basidiospores, see here, positive strain basidiospore and negative strain basidiospores. Such basidiospores, after falling on suitable substratum, germinate to produce monokaryotic, germinates and produces monokaryotic negatively strained, monokaryotic positively strained. Here it must be positive strain, or here must be positive strain, and here negatively strained mycelium. So it is produced such two mycelia of different strains that fuse to form secondary mycelium or dikaryotic mycelium. So the process of somatogamy formation of or two nuclei, positively strained nuclei and negatively strained nuclei, they fuse, diploidization takes place, dikaryotic cell, that is cell having two nuclei but nuclei do not fuse, such dikaryotic cell is produced. Then the formation of the secondary mycelium, which twists and forms rhizomer, and rhizomer produces basidiocarp, which is the fruiting body that comes above the soil, and this basidiocarp again bears basidia, and in such diploid basidium meiosis takes place, which results in the formation of two positively strained basidiospores and two negatively strained basidiospores, and the cycle completes. So in brief, we can see here, or we will see what is the systematic position or classification of Agaricus bisporus actually. So it belongs to kingdom fungi. Division of higher fungi, Eumycota. Subdivision Basidiomycotina. It belongs to class Hymenomycetes. Its subclass is Holobasidiomycetes. It belongs to order Agaricus, having family Agaricaceae. Its genus is Agaricus and species is a bisporus. So one can see here again how the life cycle of Agaricus is. By going through this figure, you can see diagrammatically how life cycle occur. So start from here. This is a mature basidiocarp formation of the basidium, and in which there is a formation of basidia. Here you will get formation of basidia and further developments, formation of primary mycelium or monochromatic mycelium. Two positive and negative strand mycelium come together and formation of this secondary mycelium. So this secondary mycelium under the soil it remains and responsible for formation of the button stage. Later in the development, the button grows, forms initials of what we can say stipe and pilus, and on its inner surface the gill chamber. Further development of this button stage and production of the mature basidiocarp. So like this you can go through this figure even, and I think you might have come to know how the life cycle of this button mushroom or edible button mushroom takes place. So commercially one can grow this mushroom and as a side business or main business you can do with this culture of artificially you can grow in your control conditions near to your house or in your field, or even on large scale you can develop it as a industry for the production of the button mushroom. They are sold fresh as well as dry preservation is done, and actually its nutritional value is very high. So fresh mushroom, it contains more than 51% of protein. Then many amino acids are there, then minerals, some of the organic acids, some of the vitamins, and all these nutritional values of button mushroom makes it a food of vegetarians, or flesh of vegetarians you can call it as a. The value, nutritional value is as good as flesh, and that's why it is called as a flesh of vegetarians. So this is the type study, we can say one of the example of Agaricus bisporus we have studied here as a representative member of group fungi. Okay.